Today, I'm going to be starting a series of 6 lectures that represent the diversity of vertebrate life and that try to add onto that backbone of diversity information about structure and function, particularly in the form of improvements or innovations that organisms are making as they move along the phylogenetic tree. Today, like I said, we're going to start with fish. Now, from the first perspective, we're all fish because we're all evolutionarily derived from fish. Fishes themselves represent a non-monophyletic grouping of organisms that have a whole bunch of non-fishes branching off of them. Those become the amphibians, reptiles, birds, mammals, and us, of course. But today we're going to focus on the entire phylogeny, but then talk specifically about that group that we colloquially or popularly call fish. Now, of course, this is nested within this larger picture of the development of deuterostomes. Deuterostomes, of course, include the echinoderms, hemichordata, xenoturbellida, and the chordates, where we're going to focus. I put X's through hemichordata and xenoturbellida because we're not really going to talk about them in this class. Those are for more specialized classes such as Animal Diversity, Biology 305. So what are some of the special things about chordates? Well, logically by their name, they have a nerve cord that transmits a lot of electric signals for communication among parts of the body. We, of course, have the vertebral column. Embedded within part of it, we have our spinal cord. These are chordates. Here are some of the branches within the chordates that represent the different shared unique derived features or synapomorphies or synapomorphies that represent the things that that entire group has and all of the descendants in that group have, at least from an evolutionary perspective, and sometimes only seen in the developmental forms. All of the deuterostomes have pharyngeal gill slits, and I'll discuss what pharyngeal gill slits are in a moment. So these are characteristics that are unique to chordates, that is, all chordates have them. They might only be visible or evident in developmental stages, or they might have subsequently lost them through evolutionary time, but they all had them at one point in time. Now those include the dorsal hollow nerve cord, which is this bundle of nerve fibers moving along the back of the animal. You also have the notochord below that, and that is this rigid structure that is used to provide a rigidity to the body, which then muscles can pull against, which enable it to have improved forms of locomotion and more rapid locomotion. They also have this post-anal tail, which is that after the anus, beyond that, you see a tail that has muscular fibers in it that is used primarily for locomotion. Now, I'm also pointing out on here the pharyngeal gill slits. These are things that all chordates have and that will be important for generating some of the things that are a critical part of chordate life. So let's talk a bit about these pharyngeal gill slits then. So they're located on the side of the head. They're an ancestral trait within all deuterostomes. They've then subsequently lost in echinoderms, which was the point I was making, that sometimes these traits that have evolved, that all of the members of the group have, will subsequently be lost through evolutionary time. In vertebrates, these pharyngeal tissues are supported by arches, that is, usually cartilaginous arches or bony arches that basically support gills that might be coming off those gill arches. If you look at the picture down here, you can see that it's showing a human embryo and some of the features of the human embryo that are sort of these developmental characteristics that humans have subsequently lost through evolutionary time, and you can only see them in the developmental stages. For instance, you can see the tail right there, and you can also see pharyngeal gill slits on the side of the human. So you can see all these vestiges of fishy ancestry in our current anatomy, and a really good book that describes some of these vestiges is called Your Inner Fish. And the book also describes the discovery of one of the early vertebrates that moved out of water onto land, which we'll return to in the subsequent lecture about amphibians. There's also a PBS special made from Your Inner Fish. It's cool. So in cephalochordates and urochordates, these pharyngeal gill slits, or sometimes called pouches, are used for filter feeding. That is, food will go in the oral mouth, and then they'll move out through the gill slits, and as they're doing, they'll be essentially filtered out so that you can take the food that's coming in with the water, let the water go out and retain the food. So this is a cool video of filter feeding in the lancelet or amphioxus, which is one of the cephalochordates. And so in this case, the scientists have given it fluorescent beads so they can track the movement of food particles through the organism. So food is entering at the front end of the animal, up at the top, and then it is being filtered out and going down the digestive tract. You can see the line of bright beads along the back of the animal. But then some are escaping out through the slits because they can't filter out everything perfectly. Now, they've subsequently been co-opted for respiration in vertebrates. So in vertebrates then, as water comes in the mouth, it passes through those pharyngeal gill slits, and then you have gills along those gill slits through which you have oxygen and carbon dioxide exchange. And now we're going to go in the branch that is specific to vertebrates. So if you take that branch that said vertebrates and you just expand it, you see a bunch of the other groups that we'd like to talk about and that we will tell you about in this course. And they're listed here. All of the things below the blue line are the vertebrates. Okay, so returning to the pharyngeal gill slits, in vertebrates these do become the gill slits. And they're bars that are between the gill slits that are called arches, as I've already mentioned, and they're formed of bone or cartilage, and then they have the gill filaments that are on the arches. So in this figure here, you can see a gill arch that has been removed with the gills on it, and this inner part here that represents the gill arch. The blue arrow is pointing to it, and the gills are then circled in red coming off of the gill arch. And on the picture on the left, you can see, looking in the back of the operculum of the fish, you can see the gill slits and the gills on the inside. So that's where the water would be coming out. So here are just two representations of the water going in the mouth of the fish and out through the gill arches, and as it goes out through the gill arches, it passes across the gills themselves, and therefore you can have oxygen exchange and carbon dioxide exchange, as well as other things across those gills. Now, the gills themselves are highly specialized for the transfer of oxygen and carbon dioxide. So on the left, on the top there, you have the different gill arches, which are going down like this with the gill filaments coming off of them. And you can see blood vessels that are going down through the gill arch that are then going out into the gills themselves. Now, each of those long finger-like filaments coming off looks like the figure in the middle. And on that one, what you have is that as the water is passing across the gill arch in one direction, the blood is moving across the gill arch in the other direction. And that is going to be optimal for the exchange of oxygen with the water. So there are capillary beds that are in these lamellae, or these gill filaments, that are for gas exchange. And the filament epithelium, that is the skin of the filament, is only 1 to 2 microns thick. And that is why you can have good exchange of oxygen and other gases and other things between the blood and the water, but also why they're very easily damaged. Now, this blood flowing and water in different directions, as opposing directions, has a really important feature and property in being able to extract oxygen from the water and being able to give up carbon dioxide from your blood. And that's called the countercurrent exchange system. Now, countercurrent exchange systems are common in many different systems. Biological ones, you'll see this repeated many times, including in the kidney, for example. There's also countercurrent heat exchange that's occurring in things like air conditioners. Now, the key element of countercurrent exchange is that blood and water are flowing in opposite directions. And what this does is ensures that the partial pressure gradient, that is the difference in gradient between the two things that are going to be exchanging gases, are maintained. And so this should change maximum exchange of gases. Okay, so why does this countercurrent exchange work so well? Well, to be able to illustrate that, we can contrast it with concurrent flow, where the blood and the water would be flowing in the same direction. So this diagram here illustrates that, where you have both the water and the blood coming across the lamellae in the same direction from left to right. So the water starts at high oxygen, the blood starts at low oxygen. oxygen diffuses from the water into the blood, and as it does so, it eventually equalizes the oxygen pressure on both sides of that exchange. That means that you can only, with concurrent movement of blood and water, achieve an equal oxygen concentration across that gradient. But now let's have the water and the blood flow in opposite directions. And if you do that, you have the high oxygenated water coming in from the left and the blood with essentially no oxygen coming from the right. And as you can see, as you move, oxygen goes from the water into the blood, but because of the countercurrent exchange, you can continue to move oxygen out of the water and into the blood across that entire gradient. So think about this. The blood comes in with no oxygen, it gets a bit from the water coming in, it moves further along, but now the water has more oxygen because it hasn't all been removed yet, so it keeps moving into the blood. The blood has more oxygen as it continues to move, but the water it's now encountering has even more oxygen because it hasn't all been removed yet, and so it continues until you can fully saturate the blood and remove most of the oxygen from the water. But then you've got that oxygen in your gills and you need to move it around the body. So this is the fish circulatory system, and I present this because it's really fun to do a comparison of hearts and circulatory systems across the different vertebrate groups. Okay, so in fish what happens is that you have the oxygen exchange across the gills, and then the blood flows through the dorsal aorta, and then the arterial blood is distributed into the body. Then it's picked up by the venous system, moved to the heart, which then accelerates the blood back to the gills where it becomes oxygenated. Now, we're going to return to this later because in some respects this is not a very efficient circulatory system, because once the blood moves through the gills, it can't be then re-accelerated to the rest of the body, which is different from us. So that is, we have two systems. One is the pulmonary circuit, that is, it moves from the heart to the lungs and then back to the heart, and then the systemic circuit where different part of the heart pumps it to the rest of the body and then back to the heart. So these two circuits then are a more efficient way of powering the movement of blood through the body. Now, so let's go away from that more efficient system that we have, but we're going to talk about how during the evolutionary lineage you see various changes in the structure of the heart and circulatory system. But now let's go back to the fish. You have the sinus venosus, where blood is coming in from the rest of the body, and then it goes into the atrium, which contracts it into the ventricle. Then it goes out through the bulbus arteriosus and to the lungs. So there's a strong muscular pump, which everything from fishes and in the fish group and all vertebrates have, that is ventrally located, so in the bottom of the body, and it maintains this blood flow with these two chambers. Now, an important feature, of course, is that you have one-way valves through each of these things, which means that as the heart contracts, the blood doesn't flow backwards. Okay, so just one other thing to tell you about vertebrates, and that, of course, is probably the most obvious thing, is that they have vertebral column, which is where the name comes from, of course. Now, this vertebral column is broken into several units. The first is the axial skeleton, which represents the head, the spinal cord, and the ribs coming off that. And then you have the appendicular skeleton, which represents the pectoral girdle, that is your shoulders, and your then they have an appendicular skeleton, which represents the pectoral girdle, that is your hips, and then you have an appendicular skeleton, which represents the pectoral girdle, it's called, which are the shoulders, and the pelvic girdle, which are the hips. And then the bones that come off that, which are the bones in our arms and our legs. Okay, and so that's all I can tell you about the general structure and innovations of fishes that represent those that are shared by the entire vertebrate group. In the second part of the lecture that we'll have in the next segment, I'm going to talk to you about the diversity of fishes, which is one of my favorite topics.